Okay, so welcome back to this next video in which we are discussing Huntington's disease and Huntington's disease medications. Okay, right, so we're currently racing towards an understanding of why Huntington's disease leads to firstly chorea in the early stages and then hypokinesia in the later stages. Okay, and it's going to be because of degeneration that's going to be occurring in the dorsal striatum. Now, in order to understand what degeneration in the dorsal striatum is going to do, we need to have a very good understanding of the basal ganglia and their function in the motor system, okay, which is what we're doing at the moment. So, we've just looked at the direct pathway, which is this pathway by which a portion of the secondary motor cortex, which has devised a motor plan that actually needs to be initiated, can send signals down to the dorsal striatum, okay, certain portions of the dorsal striatum, and activate that certain portion of the dorsal striatum. That certain portion of the dorsal striatum, which corresponds to this portion of secondary motor cortex, will then send GABAergic inhibitory inputs into certain areas of the internal globus pallidus and specific areas of the substantia nigra pars reticulata as well. Okay, and these specific areas of these two nuclei will be the areas which inhibit specific portions of the ventro-anterior and ventrolateral thalamic nuclei, which then give permission to this specific portion of secondary motor cortex. So by inhibiting those specific areas in the internal globus pallidus and substantia nigra pars reticulata, you stop the inhibition on the neurons in that area of the ventro-anterior ventrolateral thalamic nuclei, which give permission to this portion of secondary motor cortex. They then fire and give permission to that portion of the secondary motor cortex. Okay, right, so that's how you get permission. Now, what we want to look at is the indirect pathway, and the indirect pathway is all about making sure that other portions of the secondary motor cortex absolutely do not get permission. Okay, right, so let me show you this. So once again, I'll start by drawing my cerebral cortex from the left hand side, and I'm just wondering, will I have space here? I don't think I will. I'm going to get another piece of paper to do this on. Okay, so we'll start fresh here. Okay, so we're now going to see then the indirect pathway. Okay, so we'll start off once again by drawing our left cerebral hemisphere from the left hand side. So here it is. Here's the temporal lobe, and here is the rest of the left hemisphere. Right, then we've got the central sulcus, that's the first thing we always mark on, and now I'll put on the essential portions of the motor cortex. Okay, so here is the primary motor cortex, here is the supplementary motor area, a key part of the secondary motor cortex. Then we've got the premotor cortex here in blue. Okay, then in front of the secondary motor cortex here in turquoise, we've got the dorsolateral prefrontal cortex. Okay, right, so starting off again, we'll say that the dorsolateral prefrontal cortex will take the executive decision, which is, we're making this movement. It will then send that instruction to the secondary motor cortex, which will devise a motor plan. And we'll say this area is the area that devises this motor plan for this movement that the dorsolateral prefrontal cortex has ordered. Okay? We know that this area of secondary motor cortex will then send down excitatory projections into the dorsal striatum, into a specific area of the dorsal striatum. Okay, and once again I'll stress that even though I'm only showing one axon here, in reality there'll be a whole bundle of them coming down from this area, okay, of the secondary motor cortex into a specific area of the dorsal striatum here. Okay, and this is the same area of the dorsal striatum that we saw previously when we were looking at the direct pathway. Okay, now what we're going to look at is the other type of neurons that they're going to activate in here, which are going to be the indirect pathway neurons. So now you're going to activate loads of indirect pathway neurons in this area. So let's say this is an indirect pathway neuron. And the indirect pathway neurons are going to go into the external globus pallidus, shown here. So let's colour these bits in. So in orange here, we've got the dorsal striatum, or rather the putamen, which is representing the dorsal striatum. and then we've got the external globus pallidus here in red, okay, and now a whole bunch of neurons from the dorsal striatum are going to be coming in from this specific area of the dorsal striatum into a specific area of the external globus pallidus. Okay, now these neurons are once again GABAergic neurons. Okay, so they're going to be inhibitory. Okay, now what are they going to do in here? Well, they're going to inhibit the neurons in that certain area of the external globus pallidus. Now, the neurons of the external globus pallidus, they themselves are inhibitory, and they're going to be innervating the subthalamic nuclei, which we haven't previously shown on this picture, so I'll put the subthalamic nuclei here. Okay, now we're only going to need the left subthalamic nucleus, okay, because remember we're studying everything on the left hand side. So here is the left subthalamic nucleus, the STN. Okay, so you're inhibiting then an area of the external globus pallidus, and this area of the external globus pallidus will be innervating a certain area of the subthalamic nucleus. Now, these neurons in this specific area of the external globus pallidus, these are going to be inhibitory neurons, okay? In fact, the neurons of the external globus pallidus are all inhibitory neurons. Okay, so, going over this again, the secondary motor cortex is going to send activatory signals down to a certain area of the dorsal striatum. That is going to activate the neurons in that certain area of the dorsal striatum. They're going to go into a specific area of the external globus pallidus, and they're going to be inhibitory neurons. So they're going to inhibit the neurons in this certain area of the external globus pallidus. So these neurons in that certain area of the external globus pallidus are no longer going to be functioning. So these have stopped functioning. Now, what would they usually be doing? Well, they'd usually be inhibiting a certain area of the subthalamic nucleus. Okay, now what does the subthalamic nucleus do? Well, it contains excitatory neurons that are going to be innervating the internal globus pallidus and also the substantia nigra pars reticulata. Okay, so if I draw this again, here is the midbrain. Okay, like so. Here is the substantia nigra on the left hand side here, and then we've got again the substantia nigra pars reticulata. Okay, so the subthalamic nucleus will have another neuron going off into the substantia nigra pars reticulata over here, the SNPR here, and then here the internal globus pallidus. Okay, right, and these will be excitatory neurons. Okay, so if I colour these in pink, okay, and then they're going to be activating the internal globus pallidus and the substantia nigra pars reticulata. Okay, so just now to finish the picture, and then I'll make sure that the picture of this pathway is complete. Okay, so I'm going to finish the picture and then I'll explain it in more detail. Okay, so then we've got the thalamus here, okay, with the internal medullary lamina here, okay, and then we've got our two thalamic nuclei here, the ventroanterior and the ventrolateral. So here's the ventroanterior and here's the ventrolateral. Okay, right, so let's go through this pathway in full then. So, this portion of the secondary motor cortex will activate a specific area of the dorsal striatum. Okay, in this specific area of the dorsal striatum, you've got direct pathway neurons, which we've discussed, but you've also got these indirect pathway neurons. Okay, the indirect pathway neurons from this specific area of dorsal striatum are now going to be activated. Okay, and they're going to go into a specific area of the external globus pallidus, and they are inhibitory. So they will inhibit the neurons in that specific area of the external globus pallidus. Okay, now what were those neurons in that specific area of the external globus pallidus previously doing? Well, they were inhibiting neurons in a specific area of the subthalamic nucleus. Okay, now that you have lost their function, then neurons in the subthalamic nucleus in this specific area, okay, which was innervated by the specific area of the external globus pallidus, these are now going to activate. Now, what do? Well, they are going to go to a huge number of places in the internal globus pallidus and also the substantia nigra pars reticulata, and they're going to activate them. Now, remember what the internal globus pallidus and the substantia nigra pars reticulata do. They send inhibitory input into the ventroanterior and the ventrolateral thalamic nucleus. So you are activating the inhibitors, basically. So this seems like a little bit, you know, opposite to what we wanted to do in the direct pathway, until we remember what we were meant to be doing in the indirect pathway. We were meant to be making absolute sure that you did not give permission to all of the portions of the secondary motor cortex apart from the area which actually has the motor plan that we wanted to initiate. So increasing the inhibitory signal is exactly what we want to do. So basically, this area of the subthalamic nucleus that's going to be activated, this is going to activate all areas of the internal globus pallidus and the substantia nigra pars reticulata apart from the areas of the internal globus pallidus and substantia nigra pars reticulata, which then send inhibitory inputs onto a specific area of the ventroanterior and ventrolateral thalamic nucleus, which is responsible for giving permission to this specific area of secondary motor cortex. Okay, so basically you're going to activate all portions of the internal globus pallidus and substantia nigra pars reticulata which inhibit the permissive signal going to all the other areas of the secondary motor cortex, apart from the area that you actually want the permissive signal to go to. Okay, so basically the direct pathway, which we've just seen here, is going to inhibit a specific area of the internal globus pallidus. Let's say it's this area here, and it's going to inhibit a specific area of the substantia nigra pars reticulata, and I think I might draw this out again to make it clearer. Okay, so if this is the substantia nigra pars reticulata here, and this is the internal globus pallidus, you're going to inhibit specific areas of them, let's say this area and this area, and these are the areas corresponding to that area of the secondary motor cortex which has the motor plan which we want to initiate. And by inhibiting these, you stop the inhibition of a specific area in the ventroanterior ventrolateral thalamic nuclei, which will give permission to that area of secondary motor cortex. What the indirect pathway is going to do is it's going to activate all the other portions of the internal globus pallidus and the substantia nigra pars reticulata, so all of the portions that are now colouring in red. Okay, and by activating all of those portions, you increase the activity of these portions, and you increase the inhibition then of all the other areas of the ventroanterior and ventrolateral thalamic nuclei, which give permission to the other areas of secondary motor cortex, and thereby you stop the permissive signal going to those other areas of secondary motor cortex. Okay, so that's what this is all about. Let me now put one of these permissive thalamocortical neurons, okay, going to some other portion of secondary motor cortex up here, well, down here, let's say. Okay, and you're now going to have inhibited that pathway so that that now cannot give permission to that other area of secondary motor cortex. Okay, so that's what the indirect pathway is all about, stopping permission from being given to the other areas of secondary motor cortex which have got these junk motor plans within them. Okay, right. So then, that's the function of the basal ganglia. There is one final bit that we haven't actually seen, the substantia nigra pars compacta. Okay, we haven't seen the function of the nigrostriatal tract. Okay, so basically, the nigrostriatal tract releases dopamine at a tonic level into the dorsal striatum. Okay, as we saw when we discussed the nigrostriatal tract here. So these neurons in the substantia nigra pars compacta, they are firing at a tonic rate all the time and releasing dopamine onto neurons in the dorsal striatum all the time. Okay, so what is the function of this? Well, this controls how easy it is to make movements. Okay, so it's tonic, and it controls broadly how easy it is to make movements. Okay, so you know, sometimes it's really, really easy to move. It's really, really easy to go out and jog. And then other times it's really an effort to move. Okay, so if you're very, very ill, then it becomes a real effort to move. Okay, and that maybe is because you've got less dopamine in your dorsal striatum. Okay, so dopamine determines how easy it is to make voluntary movements. It is to move. Okay, now let me discuss this in more detail by discussing the actual synapses that dopaminergic neurons make with neurons in the dorsal striatum. Okay, so let's study these neurons in the dorsal striatum in more detail. Now, we've seen two different types of neurons in the dorsal striatum. We've seen the indirect pathway neurons and the direct pathway neurons, and they have very different functions, but they are both GABAergic, and in fact they have a similar histological appearance. Okay, so we're going to discuss them together. Okay, now they have a very specific name. The neurons of the dorsal striatum which release GABA are known as medium spiny neurons, or for short they're called MSNs. Okay, for medium spiny neurons. Okay, now let me draw a picture of a medium spiny neuron so that I can show you where the dopaminergic neurons are going to synapse with them. Okay, so here is a dendrite then of a medium spiny neuron. We'll have another dendrite here. Then we'll have our axon coming off down here. Okay, so this is the axon of this medium spiny neuron. And then we'll have another dendrite going off over here. Okay, oh, and I need to leave a little gap there for the final thing that I want to show. Right, so these then are the dendrites of this medium spiny neuron. Okay, this is the axon of the medium spiny neuron, which will end with an axon terminal which will release GABA. Okay, here, which will be going into either the internal globus pallidus or the substantia nigra pars reticulata if it's a direct pathway medium spiny neuron, or it will be going into the external globus pallidus if it's an indirect pathway medium spiny neuron. Okay, right, now the reason they're called medium is simply because they're medium size. Why are they called spiny? Well, this is because the dendrites are covered in dendritic spines. So dendritic spine is a little process that looks like this, that comes off dendrites. Okay, and this is the process which is involved in synapses. So when you have an axon terminal from another neuron synapsing on your medium spiny neuron, it will synapse onto a dendritic spine. Okay, so the medium spiny neurons are called medium spiny neurons because they are covered, absolutely covered in dendritic spines. Okay, now, the excitatory input that comes down from the secondary motor cortex is going to synapse like this head-on to a dendritic spine on the medium spiny neuron. So remember, excitatory inputs are coming down from the secondary motor cortex to this particular area of the dorsal striatum, and they're exciting these medium spiny neurons in that specific area of the dorsal striatum, both of the direct pathway and of the indirect pathway. Okay, right, so these will be glutamatergic synapses. They will be releasing glutamate onto the dendritic spine. Now let me show you where the dopaminergic neurons synapse onto the medium spiny neurons. Okay, so these are the dopaminergic neurons coming from the substantia nigra pars compacta here in blue. Okay, they are going to be synapsing onto the necks of the dendritic spines, and it's difficult to show because the neck's so small, but they'll be synapsing onto that little bit in here. Okay, this little thin bit here, this is known as the neck of the dendritic spine. Okay, and this is where these dopaminergic neurons, which I'll highlight in blue here, are going to be synapsing onto these medium spiny neurons. So they're going to be releasing dopamine onto the neck of the dendritic spine. Now, what does the dopamine do then? Well, this is going to depend on whether you are a direct pathway neuron or an indirect pathway neuron. Okay, so let me just discuss receptors for dopamine then. Okay, so dopamine receptors. So dopamine receptors are not going to be ligand-gated ion channels. The receptors for glutamate and GABA, okay, those are going to be ligand-gated ion channels, okay? They're going to open and allow ions into the cytoplasm of the cell. Dopamine receptors are G-protein coupled receptors, okay, GPCRs, okay, which is a huge, great family of receptors in humans. There's over 800 different GPCRs in humans. Now let me just outline for you the basic structure of a GPCR, okay. All G-protein coupled receptors have the same basic structure, which is that they are a single polypeptide, Okay, so they're a single protein effectively, okay, which contrasts them to the ligand-gated ion channels, which are multi-protein complexes. The ligand-gated ion channels consist of multiple proteins stuck together. Okay, the G protein coupled receptors are a single polypeptide. Now, the characteristic feature that all GPCRs share is that they have 7 membrane-spanning alpha helices. Okay, so if this line represents the polypeptide, they're going to span the membrane 7 times, so that's 5, and then you've got 7. Okay, so these points where you're crossing the cell membrane, and these two lines, by the way, these represent the inner and outer leaflets of the cell membrane. So this is the inner leaflet of the cell membrane, this is the outer leaflet of the cell membrane. Okay, and these areas where you are crossing the cell membrane, they will really be alpha helices. Okay, I'm just too lazy to draw them as actual spring-like shaped things. Okay, so 7 membrane-spanning alpha helices. That's the key feature of all G protein coupled receptors. Okay, so all dopamine receptors are G protein coupled receptors, so they all have this basic structure, and they're going to activate metabolic pathways inside the cell rather than ionic pathways. Okay, so they're not going to cause changes in the ion flow across the cell membrane. Instead, they're going to activate all sorts of protein cascades inside the cell, which are going to affect the cell that way. Okay, right. So there are 5 different dopamine receptors in humans, and they're cunningly labelled D1 for dopamine receptor 1, D2, D3, D4, and D5. Okay, so that's very nice. Now they're broadly categorized into 2 separate sets. Okay, firstly, D1 and D5 are put into a set. Okay, and this is the D1 type dopamine receptors. Okay, now those activate GS heterotrimeric G proteins. Okay, so they activate a certain downstream signaling pathway. Okay, within the cell. Okay, so both D1 and D5 do the same thing effectively. They activate the same signaling pathway within the cell. Then D2, D3, and D4, they activate a different signaling pathway to D1 and D5. Okay, but they all again activate the same pathway together. So they are known as the D2 type dopamine receptors, and they activate GI heterotrimeric G proteins. Okay, so those are the D2 type dopamine receptors. Okay, right. Now, why have we gone into this? Well, basically, the direct pathway medium spiny neurons. So remember, the medium spiny neurons that are going to be sending their axons into either the internal globus pallidus or the substantia nigra pars reticulata, they are going to have D1 receptors, okay, on their necks of their dendritic spines, okay, whereas the indirect pathway medium spiny neurons are going to have D2 receptors on their dendritic spine necks. Okay, so dopamine is going to have a very different effect on the direct pathway neurons compared to the indirect pathway neurons. Okay, so now let me explain what dopamine is going to do to these 2 pathways. Okay, so we'll start with the direct pathway. So the direct pathway medium spiny neurons, now I'll get the direct pathway back up here, they have got the D1 receptor on their dendritic spine necks. Okay, so when dopamine is released onto that D1 receptor, what it will do is it will increase the sensitivity of that dendritic spine to the glutamate. Okay, so what do I mean by that? Well, when glutamate is released from the cerebral cortex neuron onto this dendritic spine, what will happen is the glutamate will open glutamate receptors, which are ligand-gated ion channels, and these ligand-gated ion channels will then allow in a current of sodium ions into the cell. Okay, so in come the sodium ions, and those sodium ions will make it more likely then that the medium spiny neuron will fire an action potential. Okay, basically, if the dopaminergic neuron is releasing dopamine, a lot of dopamine, onto the neck of the dendritic spine, that dopamine will be activating D1 receptors on the neck of the dendritic spine, which are coupled to the GS, the stimulatory G protein, okay, and that's going to result in this dendritic spine now becoming more sensitive to glutamate. And literally what that means is that every time this cortical neuron releases glutamate onto the dendritic spine, the amount of sodium current, okay, that you're going to get coming into the cell is going to be bigger, okay, because of the dopamine. So it's going to make the sodium current that comes in due to the glutamate bigger, basically. So it's going to make this neuron more excitable, okay? So it's going to up the activity of the direct pathway. So the direct pathway is going to go up because of dopamine. Now, what does that mean? It means that it's going to be easier to initiate voluntary movements, okay? Because remember, the direct pathway is the pathway that gives you permission to send this motor plan onto the primary motor cortex. So dopamine, by activating the direct pathway, is going to make it easier to initiate voluntary movements. In addition, what is dopamine going to do on the indirect pathway? Well, dopamine is going to activate D2 receptors on the indirect pathway neurons. Now, what these are going to do is they're going to desensitize the indirect pathway medium spiny neurons to glutamate. So remember, the indirect pathway medium spiny neurons here, they will also have cortical projections releasing glutamate onto them. Now, when the dopaminergic neurons are releasing lots of dopamine onto the dendritic spine next of these indirect pathway medium spiny neurons, that dopamine will be activating D2 receptors, and those D2 receptors are coupled to the GI, the inhibitory heterotrimeric G protein, and that's going to desensitize the dendritic spine to the glutamate now. So, when this cortical neuron releases glutamate onto the dendritic spine, the amount of sodium current that you're going to get coming in is going to be reduced. So it's going to make the indirect pathway medium spiny neurons harder to activate. Okay, so you're now going to reduce the activity of the indirect pathway. Again, this is pro-movement, because remember, the indirect pathway is stopping permission from going to the other areas of the secondary motor cortex. So by reducing its activity, you're effectively making it easier to give permission to other areas of the secondary motor cortex. So again, this is helping you make movement. Okay, the more dopamine you have, the easier it's going to be to make movements, basically. Okay, of course, if you have too much dopamine, then you're going to get problems. Okay, if you get too much dopamine, you're going to start getting chorea, basically. You're going to be getting hyperkinesia, because motor plans in the secondary motor cortex that are not supposed to be initiated are now going to be initiated because the indirect pathway is too low in activity. Okay, so it's about having a healthy balance, basically. If you end up with too little dopamine, which is what happens in Parkinson's disease, then the direct pathway becomes too inactive, the indirect pathway becomes too overactive, and movement stops, basically. You can't really struggle to initiate voluntary movements. Okay, so it's about having a balance there. Okay, right, so that's what dopamine's role in this system is. It is a tonic signal that determines how easy it is to make movements. The more dopamine you have, the more easier it is to make movements. The less dopamine you have, the harder it becomes to make movements. Okay, and this will vary in physiology. Okay, in pathology it varies extremely, so you end up getting dopamine levels that are far too high, and that causes over movements. Okay, so hyperkinesia. And if you end up with far too low dopamine, as happens in Parkinson's disease, you end up with far too few movements. Okay, movement becomes really difficult. Okay, so that finishes then the physiology of the basal ganglia system. In the next video, what we will do is we'll see what happens in Huntington's disease. We'll finally see which neurons are going to degenerate in Huntington's disease. Okay, and we'll see how this causes those motor symptoms of the early and later stages of Huntington's disease. We'll then move on to the drug treatment of Huntington's disease.